Good morning, everybody. Now for something completely different. Talk about intimidating. To follow two amazing speakers. Well, so anyway, I am a research scientist, and I'm also an alum. Actually, I got my PhD here in biology, molecular developmental biology, and now I'm faculty at Tufts. So I'd rejoined Tufts about 6 years ago, and I'll tell you a little bit about we're doing something completely different. This is science on a Saturday morning. All right, but here we go. So basically one of the things that we're doing is working on dental tissue regeneration. We're actually working on mineralized tissue development and regeneration. And when I joined the faculty, actually, at Tufts, one of the things we had been working on for a while was tooth tissue engineering. And so why do that? I just love learning about Tisch. I really have to say that I really did not know much about Tufts Tisch at all. And one of the things that we're really interested in is actually improving oral health because it's so important for all sorts of aspects of life. And when I started working in this field, I was actually shocked to learn some of these statistics, like the fact that 28% of all preschoolers have tooth decay. And this is in the United States, so this is in a wealthy community, well-educated community. And greater than 50% of children aged 6 to 8 have tooth decay, and by 17, approximately 80%, greater than 80% have already lost teeth. You can read the statistics. It just gets worse, and so that by age 70, approximately 50% of the population is edentulous, and this is a real problem with eating, with social interactions, with just overall health. And also long-term denture use can result in bone resorption, congenital tooth loss, cancer and trauma, particularly now in this era of way too many wars where body armor is improved, but the face needs to be opened for seeing and hearing. And it's a very expensive endeavor to do replacement teeth. It's about $2,000 a tooth or $25,000 for a jaw. It's a huge industry. But it's really the only option that you have is to use these titanium implants. And many people aren't eligible for dental implants based on bone resorption or the fact that they can't afford them. And if you look down here, this is in 2005, the statistic really shocked me that there are about 5 million implants that were placed, and if you estimate a 97% success rate, which is actually very optimistic, that means that there was a failure every 4 minutes. So this is a real problem. And so, and bioengineered tissues, so certainly I'm not the only person working on teeth, bioengineered teeth or bioengineered tissues in general. All of us in the tissue engineering field are hoping to make organs that are every bit as good, if not better, than the organ that they're trying to replace. And for teeth, you can read what we're looking for, just better integration. So instead of having a cavity filled by a metal, that you can have microseepage and having the cavity, the filling fall out, if you could actually regrow those tissues and actually have your dentin and enamel crystal structures growing. So I'm going to tell you a few things, three things. One is that I'm going to try to talk about our basic and translational dental research. I'm going to give you some examples of our mineralized tissue regeneration, and then lastly, I'm going to introduce you to the zebrafish model for those of you who don't know the fish, if I get to it in 10 minutes. So I have two laboratories. I have a laboratory downtown in Boston, which is our zebrafish group, where we have a 2,000-tank fish facility for using the zebrafish to study human mineralized tissue diseases. And then in Medford, I have our dental tissue engineering lab over here down at 200 Boston Ave, where we're working with other engineers that are all over the campus around here to try to improve our dental tissue engineering applications. So basic tooth development. I'm not going to do this I'm not going to be able to do this in 10 minutes. But just let me tell you that tooth development is very complex. I had no idea when I started this how confusing it was going to be, but it starts with just the interactions of these two tissues and then results in this really highly mineralized organ that then erupts. So not only do you have your soft tissue development, which most soft tissue organs do, you also have to mineralize afterwards. But the mineralization is based on the soft tissue template. And so I love this quote from Jay Vicanti, who got me into dental tissue engineering. So he's chief of pediatric surgery at Mass General Hospital. And he and Bob Langer, a brilliant bioengineer over at MIT, believed that the most efficient road to human therapy and regenerative medicine and tissue engineering is combining biology with technology. And that's exactly what everybody in this field of tissue engineering are doing, regenerative medicine. So what we did, based on the successful applications of many other people, was taking these immature tooth buds, dissociated the cells, expanding them in culture, seeding them on a scaffold because the cells don't like to be hanging out by themselves, and then implanting them in a host to give it a vasculature, which is really key, and then seeing what happens. So here's a pig tooth bud, here's a scaffold. Here's what we got. And I hope you can appreciate from that really fast red and green picture that these cells reorganized themselves without any additional influence. We just seeded them together. They reorganized themselves into these beautiful structures that look just like naturally forming teeth. So that was really fun. And we did this with rat. We got a lot of publicity, the front page of the Globe. We were on the news. We had no idea what we were doing. And we were just really thrilled to see what was happening. So we know tissue engineering can work for teeth, but we don't know how to do this. We don't know how to generate, if you lose a tooth, we don't know how to replace it yet. We don't know how to replace it with a tooth that actually matches your mouth. And so we're trying a variety of different things. Now this doesn't count. I get a few seconds extra because this isn't, there it goes. Oh no. Oh no. All right, so let me go back real quickly. So what we're doing is using human tooth bud cells. Now that I moved to Tufts, we get lots and lots of extracted teeth every week from the Department of Oral and Maxillofacial Surgery, and we're taking the cells that are resident in these wisdom teeth or other extracted teeth, and we're culturing them, expanding them. These are stem cells. They can differentiate into different cell types, and we're taking periodontal ligament tissues as well, expanding those in culture, and we're combining them with biodegradable scaffold materials such as silk, and we've shown that these cells can form structures that adopt the pore sizes of these silk materials, so potentially could be used to develop roots. We're trying new hydrogel materials. There's lots of new nanofabricated materials that I think you'll hear more of later this morning to try to generate two structures showing cell-cell interactions. And we're also trying cell sheet technologies where you can actually grow a layer of dental epithelial cells, a layer of dental mesenchymal cells, put them together, and hopefully form a tooth right over here. We're working on implants. We're working on bioengineering periodontal ligament tissues onto titanium implants, which is showing here. We're working on bone tooth constructs. We're using an autologous mini pig model where you can actually make a bioengineered tooth, attach it to a bone, and grow it in the jaw. It's working with certain success. And then the fish. Get to the point. Fish regenerate teeth throughout their lives. This is a zebrafish replacement tooth forming. This is a baby tooth, our deciduous tooth in a human, and this is an adult tooth that's forming off of the baby tooth. So zebrafish continuously do it throughout their lives. We can only do it once. Why is that? We're hoping to figure it out. And so by studying this dental stem cell niche where these replacement teeth form in the exact size and shape of the tooth that they're replacing, we're hoping to learn cues as to how we can maybe go to the dentist when our adult tooth is a little bit younger than this, generate a new replacement tooth to form, and then you'll have it. If something ever happens to our adult tooth, you could have this backup replacement tooth that would be cued to develop in response to losing the other one. So that, kind of in a nutshell, I have a minute left, is to tell you we're also using the zebrafish to study any sort of mineralized tissue diseases. The zebrafish have been used mostly to look at embryonic developmental defects, but what I'm interested in my laboratory, huge laboratory, is interested in looking at adult, viable mineralized tissue mutations that we can use to study for human disease. So there are all sorts of mineralized tissue diseases that you can think of. You probably know people that have osteoporosis or perhaps cleft lip and palate. And so we're raising these animals, and it's really laborious. It really takes a lot of time and energy. I have a wonderful laboratory that's doing all this just to identify these mutants and then identify the gene mutations that are responsible for that to try to use these animals as a model to study human disease, which you can't do in people, as we know. So I thank you very much for all of your time and help, and these are part of the team that have been working on this. It's a big crew and have had lots of support from also many funding industries and all of my collaborators as well. And I thank you for your time.